I'm Cecilia Conaco. I'm with the UP Marine Science Institute, and today I will be talking about some work that we did to explore the early evolution of the synapse. So, nervous systems are a unique feature of all animals. They allow us to sense and interact with our environment. And within the animal kingdom, you can see two main kinds of nervous system. You have the diffuse nerve net, which you see in a lot of marine organisms like corals, jellyfish, and sea anemones. And then you have the centralized nervous system that you see in invertebrates like worms and flies, as well as in vertebrates like fish, dogs, and humans. Of course, there are always exceptions, and the porphyrians, or sponges, which are also multicellular animals, do not possess any true organ systems, and they do not have a nervous system. In most animals, except for the sponges, nervous systems are composed of specialized cells, which are called neurons, and these neurons are connected to each other in very special ways to form neuronal circuits, which allow for the directional flow of information. And the transfer of information between neurons is mediated through special junctions called synapses. Neurons transmit information through both electrical and chemical signals. An electrical signal traveling through the body of one neuron will trigger the release of chemical neurotransmitters when it reaches the presynaptic terminal. These chemical neurotransmitters will then cross the synaptic cleft, and they are recognized by receptors that are found in the postsynaptic membrane. Once these receptors are bound, they will trigger another electrical signal in the second neuron, and this will continue the flow of information. The synaptic connections between neurons are highly dynamic. They are continually being formed and retracted, and as an organism develops and experiences new things, the synaptic connections or the circuitry of the nervous system becomes more complex. And the strengthening of specific connections between neurons is the basis for learning and of memory storage. As you can imagine, all these different synaptic connections have to integrate a lot of information. And because the function of the synapse is very complex, it's also no surprise that it requires a lot of different proteins in order to do its job. Each synapse has about 100 different proteins, some of which are shown in this diagram. The synapse has two main parts. You have the presynaptic compartment, which contains the proteins required for packaging and transport of chemical neurotransmitters, while the postsynaptic compartment has your neurotransmitter receptors and signaling molecules, as well as the scaffolding proteins that are required for keeping everything in place. Looking at the structure of the synapse, one just has to ask, how does nature build such a complex cellular machine? And one way in which we can think about this is to think of it in terms of how we would build any kind of multi-component machine. So for example, if I were to build a car, which would also require many different components, I would have to put all of these together in very specific ways so that I can build a functional car. So to build a complex machine, cellular or man-made, there are several steps that you would have to consider. First of all, you would need to invent the different parts, and for the synapse, this would be equivalent to inventing the different the genes, so gene innovation. The second factor that you would have to consider is that all the different parts should be able to assemble together in order to perform their function. And for the synapse, this would involve the different proteins interacting with each other in very specific ways. And finally, of course, all your different parts that make up your car or your machine have to be collected together in the same place. And when you think about building a synapse, this would involve co-expression of the different genes in the same cell or in the same neuron. And to get evidence for each of these different processes or steps in the evolution of the synapse, we can apply a method known as comparative genomics. Comparative genomics essentially means comparing the genomic content of different organisms so that we can make inferences about when certain features or genes first originated. And this has become possible now because of recent advances in sequencing technology. So now we can look at the genomes of many different organisms that represent key steps in the evolution of animals. The evolutionary relationships between different organisms can be represented in the form of a phylogenetic tree. And so for animals, if we start off at the very base of the tree, we have the Holozoan ancestor, which is the ancestor that gave rise to all multicellular animals and their closest unicellular relatives, which are called the choanoflagellates. The next node on this phylogenetic tree is the Metazoan ancestor. The Metazoan ancestor gave rise to all multicellular animals, and the earliest form is represented by the poriferans, or sponges, which do not have true organ systems, and they don't have neurons, as I mentioned earlier. But sponges or poriferans are known to have sensory cells. Next up on the tree is the Eumetazoan ancestor. This is the ancestor that gave rise to all true animals, so animals with true organ systems. And the earliest form of Eumetazoans are the cnidarians, which include your corals, jellyfish, and sea anemones. Most of the members of this family possess a diffuse nerve net. And then finally, we have the bilaterian ancestor. Bilaterians include invertebrates and vertebrates, and most of the animals in this group possess a centralized nervous system. By comparing synaptic genes in each of these different organisms, we can then infer when the different parts of the synapse first came about. So now let's go through all the different parts of the synapse and look at when each of the different genes first originated. So here is a diagram of the synapse again, with each of the proteins that emerged at the Holozoan ancestor colored in blue. And what you will see is that even at this earliest point in evolutionary time, you already see many parts of the synapse are present. So you have certain proteins in the presynaptic terminal, and you even have a scaffolding protein called PSD95 in the postsynaptic terminal, already existing at this point. But it is when you look at the metazoan ancestor when you see a majority of synaptic genes emerging. So here, you'll see that you almost have a complete set of synaptic genes, but you're just missing a certain key components. For example, you don't have certain proteins that link your synaptic vesicles to the presynaptic membrane, and you're also missing the receptors that are required for recognizing your chemical neurotransmitters in the postsynaptic membrane. Most of these missing proteins emerge in the eumetazoan ancestor. So as you can see here, the functions of the pre and the postsynaptic terminal are already linked, and you can infer that quite possibly at this stage, you can already form a functional synapse. When you look in the bilaterian ancestor, very few genes are added. So there's a few modifications that are made to the synaptic structure, but the essential core synaptic complex remains the same. So this tells us that most synaptic genes evolved way before the emergence of the first synapses or first nervous systems. And so we have to think about the other aspects that are important for building a cellular machine. And that would be the ability of the different proteins to interact with each other, and also co-expression of the different genes. So if we look at how the different synaptic genes are able to interact, in the synapse, most synaptic genes are organized by scaffolding molecules that contain PDZ domains. A PDZ domain is just a specific protein folding structure that contains a binding pocket that will recognize a specific target sequence on the end of certain genes. And in the synapse, you'll see many scaffolding proteins that have multiple PDZ domains. And what these allows the scaffolding proteins to do is to organize multiple proteins with different functionalities very efficiently into the same location. And this becomes really important when you look at the postsynaptic complex. So in the postsynaptic complex, you have the receptors that recognize neurotransmitters, and these have to be kept in close proximity to signaling molecules that continue the propagation of information. And all of these different proteins are organized by a scaffolding molecule known as PSD95. So we took a closer look at this complex that is organized by PSD95 and performed a comparative genomics approach similar to what we did for looking at the origins of synaptic genes. Instead, this time we were looking at when the specific sequence at the tail end of these proteins emerged. So to look at when the ligand sequences on the proteins that interact with PSD95 first evolved, we again used a comparative genomics approach. And what we found is that although the genes emerged at early time points, they did not necessarily evolve together with their ligand domains. This tells us that continuous modification of the sequences for pre-existing genes was required in order for these different sequences to evolve and to attain the ability to interact with the scaffolding proteins. And finally, the third step that's required in building a synaptic machine is to co-express all of the different synaptic genes in the same cells. It's generally known that genes that work together tend to have similar expression patterns over developmental time. As shown in this example, genes 1 and 2, which share very similar expression patterns, quite possibly also share function or work within the same cellular process as compared to gene 3, which has a very different expression pattern. The similarity in expression patterns for different genes can be expressed as a correlation, and this correlation can be displayed in the form of a heat map, where very high similarity in expression is shown in warm colors, like red, and very different expression patterns are shown in cooler colors, like blue. So we can do this same analysis for all the synaptic genes that are present in different species. And if we look at synaptic genes in the sponge, what we find is that there are several sets of genes that share very similar expression patterns, as you can see from the red blocks across the diagonal. But where you really see an increase in co-expression is when you look at an organism that has a functional nervous system. So for example, coral, which has a diffuse nervous system, also show a very high co-expression between all its different synaptic genes. So this is telling us that because these genes are working together, to some extent they also are already being coordinately expressed. And in fact, this increase in co-expression is even more apparent when we look at an organism that has a centralized nervous system, for example, the fly. So here is the correlation heat map for fly synaptic genes. And what you'll see is that there are two very large groups of genes that share very similar expression patterns. But even between these two groups, you also have a substantial overlap in co-expression over development. And so these results tell us that an increase in co-expression is a signature of a functional nervous system. So it's likely that as the organisms evolved, these synaptic genes became controlled by very similar factors so that they can be expressed in the same cells at the same time. To summarize what we know about the early evolution of the synapse, we now know that many synaptic components emerged way before the evolution of the first nervous systems. And these synaptic genes first emerged without the ability to interact with each other. And through continuous modification of their gene sequences, they eventually acquired the ability to interact with each other and with scaffolding proteins that are required for forming synaptic structure. We also know that rewiring of gene regulatory mechanisms must have been very important so that these different synaptic genes are then expressed in the same cells and at the same time. Clues in the genomes of early animals suggest that building a functional synapse involved gene innovation and the repurposing of pre-existing genes, modification of gene regulatory mechanisms that allowed these different genes to be expressed in more similar ways, and also the reorganization of protein interactions that allowed the core structure of the modern synapse to be formed. But of course, our understanding of how the synaptic machinery evolved and how other cellular structures evolved will only get better as we gain more understanding of the genes that are found in other members of the animal kingdom.